Et donc Igor suggérait que vous introduisiez toute la session comme il est sur Teams, c'est que c'est plus facile si c'est vous.
Pas de problème, il est assez grand pour le faire. Attends, je vais lui dire. Igor, tu peux introduire en tant que président, il n'y a pas de problème, on t'entend d'ici. Mais si tu mets ton micro, ça ira mieux.
Le son au maximum.
Vas-y un peu. Non, moi j'ai un problème. Ils ne m'entendront pas. Du associé, regarde.
Faut mettre la sortie.
Comment? 3 petits points. Device setting. Vas-y un peu.
Oui, tu m'entends?
Voilà, c'est parfait.
Ok. Attends 2 secondes, je demande qu'on fasse moins de bruit autour de moi. Et puis je reviens.
On est au bord de la plage.
Je ne suis pas au bord de la plage, mais il y a les enfants qui sont autour de moi et qui font un peu de bruit. Là. Quand Camille est prête.
Je suis prête.
Bien, donc, bonjour à tout le monde pour cette défense publique un peu particulière de Camille, étant donné que certaines personnes sont en présentiel, d'autres en distanciel. Donc nous sommes réunis aujourd'hui pour cette défense privée de Camille qui s'intitule Optimization of the Exopolysaccharide Production of Gloeteke verrucosa by Modification of the CN ratio and Investigation of Future Industrial Applications. Et donc, pour cette défense, nous avons un jury d'experts qui est composé du professeur Konrad Meulart de la KU Leuven, du professeur Philippe Michaud de l'Université de Clermont en Auvergne, France. Il y a également donc le professeur Annelise Hanson de l'Université de Mons, le professeur Rudy Wattier qui est donc le directeur de cette thèse, et moi-même Igor Ekod qui suis là en tant que président du jury. Je vais donc demander à Camille de faire sa présentation durant 40-45 minutes et ensuite donc les experts qui se sont déjà réunis et qui ont déjà posé un certain nombre de questions à Camille pourront encore poser quelques questions ainsi que l'Assemblée si elle le veut. Donc Camille, je te cède la parole pour cette présentation pendant 40-45 minutes. Merci à toi.
Merci beaucoup. Est-ce que tout le monde m'entend bien sur YouTube et dans la salle? C'est bon? Je suppose que oui, puisque Guillaume ne se relève pas. Alors, I would like first to thank all the members of the jury to be here today and all friends, family and colleagues to be here. I'm very happy to present you the result of five years of work concerning the production of exopolysaccharide Biocyanobacterium gloeotc verrucosa when we modify the carbon to nitrogen ratio and in potential use of this polymer in industries. I think that to better understand what we wanted to do during these 5 years, I need to explain you some notions which could be interesting to understand for all of us. And I would like first to introduce the selected organism, Gloeotc verrucosa. But for that, I need first to introduce the microbial world. So you all know that microbes, microorganisms, bacteria are very important for the earth. And maybe if I ask you to draw a microorganism or a bacteria, you will draw something like that. A bad, a wrong organisms which cause diseases, and it's actually more true perhaps. But it's completely false because there is a huge variety of bacteria which are very important for the development of the earth and for the ecosystem today. And among these bacteria, there is multiple metabolism. And these metabolisms can be classified according to characteristics. The first one is associated with the energy source. So what is the energy used to produce the ATP in the cell, the energetic molecule of the cell? The second characteristic is what is the building blocks associated with the metabolic constituents. So, if we focus first on the energy source, there is two possibilities. The phototrophy, when the light energy is used to produce the ATP in the cell, or the chemotrophy, when molecules are used to produce the ATP. Concerning the second characteristic, there is either autotrophy, when inorganic sources are used to constitute the different molecules in the cell, or heterotrophy, when organic sources are used. And according to these two characteristics, you have different metabolism and different organisms. And the one which interesting us today is the cyanobacteria group, here with the green color, as plants, and they are characterized by a phototrophic metabolism. So they use the light energy to produce the energy in the cell in the form of ATP, and they are autotroph because they can use the CO2 of the atmosphere to produce carbonate molecules in the cell. These cyanobacteria are very important for the development of the earth and the history of the earth. Effectively, if we go a few billion years ago, around 3 billion years ago, there is the origin of cyanobacteria in the aquatic environment. These organisms so use the energy of the light, the energy here to produce the ATP, and this part of the metabolism is associated with the formation of the oxygen. At this period of Earth development, no oxygen were present on the atmosphere. So they aim to increase the oxygen content in the environment, and associated with that, it's also associated with the formation of the ozone shield. And the increase in oxygen content and the formation of ozone shield permit the development of terrestrial organisms, and so new form of life, explaining why they are fundamental in our history. And cyanobacteria is a group of organisms which is constituted of multiple species. Some studies highlight more than 8,000 species with different morphology and different metabolism. You have, for example, unicellular organism with different shapes, such as cocoid or fusiform, but also pluricellular organism in the form of filament, which could be branch or not. And if we zoom on this filament, you can see here on the microscopy picture a succession of segments, and in fact each segment is a cell. So the filament is a succession of hundreds of cells. In our case, among all these species, we select one organism, which is a unicellular cocoid Cyanobacterium, Gloeotece verrucosa. The strain was isolated from a rice field in India, so it's a freshwater cyanobacterium, unicellular, but the strain is also interesting because it is able of a diazotroph metabolism. So in addition to use the CO2 of the atmosphere, the strain is also able to use the nitrogen, the denitrogen of the atmosphere to have a nitrogen sources. If we go deeper to analyze these organisms, we can see here the structure of the membrane. The structure of a Gloeotece verrucosa membrane is formed of two membranes, in fact, an outer membrane and an inner membrane, named plasmic membrane. And between these two, you have the periplasm formed of peptidoglycans. if we are looking at the intracellular space, you have here invagination of the membrane, which form what is named thylakoid membrane. These thylakoid membranes are associated with the photosynthesis metabolism. But you have also different kind of granules, as you can see here. You have, for example, white granules and black granules, which are polyhydroxyalkanoates or glycogen granules. These two represent a carbon reserve for the organism. And you have also gray granules, which are cyanophysin granules, representing, in this case, nitrogen reserves. And now, if we go deeper to understand all these constituents, we will start with the photosynthesis machinery present in the thylakoid membranes. So the photosynthesis is based on different membrane proteins, and among these different membrane constituents, you have two photosystems, the photosystem 1 and the photosystem 2. Here I represent the photosystem 2, and if you zoom in this photosystem, you can see represented here in different color the proteins, so in red, in blue, and in gray. And these proteins form together what we call a phycobilizome, and the proteins are named phycobiliproteins. So if we have here the light energy, this light energy will be harvested by this protein, then the energy will be transferred from protein to protein until the chlorophyll pigment, so the one which is also present in plants, and this is the chlorophyll which gives the green color to cyanobacteria. And this transfer of energy will be then associated at a larger scale to electron transferred. Effectively, we have here so the thylakoid membrane with the two photosystems, the different membrane components, and the light energy, then the transfer of electrons, which are transferred among all these different membrane components. And you have also associated with this photosystem 2 the production of oxygen. This electron transferred is very important because associated with a gradient of protons. And this gradient of protons is important because the accumulation of these protons is important for the good functioning of one protein, one enzyme, named the ATP synthase, which is associated with the production of the famous energetic molecules, the ATP. So it presents you here the transformation of the light energy in a chemical energy in the form of ATP. As I also present you just a few slides before, there is also different granules present in the cell of Gloeotece verrucosa. And if we zoom on these molecules, and if we analyze the structure of these molecules, you have here the polyhydroxyalkanoate and the glycogen granules. Both are, as we can see here in red, rich in carbon atom, so they represent the carbon reserve. And you have the cyanophysin granules here, which is rich in nitrogen atom. So why it is important for these organisms to have this kind of reserve in the cell? Because it will be important if we analyze the nutrients which are present outside the cell. So in the case of deficiency or excess of nutrients, the strain is able to constitute these different granules and degrade these granules when there is a deficiency. And carbon and nitrogen are two essential elements for the development of an organism. And it leads me to the next notion that I want to present you. This is the CN ratio, so the carbon-nitrogen ratio. But for that, I need first to introduce the notion of metabolism. So the metabolism is a succession, an accumulation of different reactions. But what is a reaction? We can briefly represent a reaction as the transformation of a compound 1 in compound 2 thanks to an enzyme. But at a cell scale, it looks like this. You have multiple reactions, and all these reactions will not occur exactly at the same time. So the cell needs to have different points of regulations. and there is different point of regulation. One of these points is the monitoring of the different elements, the different nutrients which are present outside the cell. Among these different nutrients, there is the carbon and the nitrogen, and these two elements are associated thanks to the CN ratio, the carbon to nitrogen ratio, and so their regulation is connected. Effectively, if we represented here a cell, you have highlighted in red the different nitrogen sources which could be used by Gloeotc verrucosa, so the nitrate, the ammonium, the urea, and the dinitrogen of the atmosphere. All these elements will enter the cell and will be transformed into ammonium because this is the assimilated form of nitrogen. And this ammonium will be assimilated into one amino acid named glutamine. Concerning the carbon, in blue in this case, so you have here the CO2, which will also enter the cell through the Calvin cycle, then the Krebs cycle, and at the end you will have the formation of 2-oxoglutarate. And in fact, the regulation between the carbon and the nitrogen will not be associated with the concentration present outside the cell, but with the content in glutamine and in 2-oxoglutarate. If we take an example, we are in a situation in which you have an excess of carbon and a deficiency in nitrogen. What will happen in the cell? You will have first the excess of carbon, which will be introduced in different polymers, the glycogen granules or the polyhydroxyalkanoates. But this excess of carbon is also could also be integrated into polymers, which will be secreted out of the cell. If we analyze what is the response concerning the deficiency in nitrogen, we will have the answer of the cell with this associated with an increase of the assimilation of nitrogen sources present outside, but also a degradation of cyanophysin granules, the nitrogen reserve of the cell. But it's not the two possibilities. There is also other possibilities for Gloeotc verrucosa. And another possibility is to use the phycobiliprotein present in the photosystem too. Because if you degrade these molecules, you can see that they are also rich in nitrogen, so they can represent another nitrogen reserve for the organism. So in addition to have the degradation of cyanophysin granules, you can also have a degradation of phycobiliproteins. So it gives here a brief overview of what is happening in the cell when there is a dysregulation of the carbon and nitrogen ratio. The last notion, I want to present you is the notion of exopolysaccharide, because this is the product that we want to produce during this thesis. So exopolysaccharide is the combination between extracellular and polysaccharides. So polysaccharides secreted out of the cell. But what are polysaccharides? As represented here with the blue hexagon, it's a long chain of carbohydrates composing of monosaccharides. So each hexagon is one monosaccharide or one sugar. And all these monosaccharides are connected together thanks to glycosidic linkage or glycosidic bonds. And we can define extracellular polysaccharides, so EPS, according to their composition first. So if you have a homopolysaccharide, so you have a polymer in which you have just one type of sugar inside the polymer. Or if you have heteropolysaccharide, you have multiple monosaccharides which could form the polymer. And I give you here a brief overview of different monosaccharides that you could find in exopolysaccharides. Perhaps two that you may know is the glucose because of diabetes, but also the ribose, which form our DNA. And in addition of these long chain of carbohydrate, there is also non-saccharidic constituent which are closely associated with the exopolysaccharides, for example, sulfated groups or proteins. And all these elements together are very important for the physicochemical properties of exopolysaccharides. Another possibility is to define EPS is according to the location. For example, you will have released polysaccharide or RPS, representing here by the bubble when the polysaccharide is completely free in the environment. In the second case, you can have capsular polysaccharides, or CPS, when polysaccharides are still attached to the external membrane of the cell. And if we want to have a visualization of these two fractions in glaucoma verrucosa, you have here a representation with a blue staining these exopolysaccharides, and you have here blue zone, which represent the RPS. So if you have just blue zone but without any cell, so the released polysaccharides, and you can see that around the cells here, you still have blue halo, which represent the capsular polysaccharides. So glaucoma verrucosa possess the two fractions, the RPS and the CPS, and these both fractions form the total exopolysaccharides. These exopolysaccharides are secreted out of the cell, and they have important roles for the bacterium. They have physiological roles, and among these, you have a role of protection. Yes, you can have a protection against harsh condition, for example, desiccation, UV radiation or highlight intensities, but there is also a role in the biofilm formation. So exopolysaccharides are at the basis of biofilm formation, which is important for an intra or interspecies communication. And these exopolysaccharides are important for the organisms which produce these elements, but they are also useful for us because we use exopolysaccharides in our daily life. For example, there is application of exopolysaccharides in the food sector. One example is their gelifying properties in the jam that you put on your toast every morning. There is also many applications in medicine. One example has vaccine. Exopolysaccharide actually in USA are associated with three vaccines. There is also a role in cosmetics. One example, which is a well-known example, is the hyaluronic acid that you find on anti-aging cream. This hyaluronic acid is produced by a bacterium. And there is also a role in agriculture because these polymers could replace the different fertilizer used today. And because we use these polymers in multiple sectors, actually, we need to have organisms able to produce a huge amount of these exopolysaccharides. So for that, we need to, at the laboratory scale, increase the production of exopolysaccharide in organisms. And it is possible, effectively. We can switch from a low content to a high content of exopolysaccharides by playing on different environmental parameters. First, we can play on the nutrients, nitrogen, carbon, sulfate, but also play on parameters such as the temperature or the light intensity. And in addition to switch from this content and to optimize the production, we can also play on the composition of the polymer and on the different monosaccharide forming the polymers, which is very important because it can change the application in industries. And one example in which we can play on is the carbon to nitrogen ratio that I presented you a few slides before. So all these elements, I think, permit to better understand what we wanted to do during this thesis. So we play with glaucoma verrucosa. We modulate the carbon to nitrogen ratio. We analyze the response of the cell, and we analyze also the exopolysaccharide production. We then try to characterize the polymer and see if it could be interesting or not for the industries. So the first part of the result will concern the response of the cell and the exopolysaccharide production, which is associated with the different CN ratio that we tested. So effectively, as I told you, we play on the carbon to nitrogen ratio. And to do that, we change nitrogen sources and concentration. So we test nitrate, ammonium, and urea at 3 different concentrations to modulate this ratio. And we also test the use of atmospheric denitrogen as sole nitrogen sources. We analyze the response of Gloeotc verrucosa by analyzing of the growth, the intracellular compound, and the exopolysaccharide production. The growth, how we can measure the growth? We analyze the growth by measure of the turbidity of the culture. So we measure the increase in optical density over the time. And if you have an increase in this optical density, you have a growth of the culture. And in this case, during the nitrogen excess condition, so one of the two profiles that we obtain, we have a growth of the strain with an OD reaching around 1. Concerning the intracellular reserve, we analyze the phycobilly proteins and the cyanophysin granules, but in addition, we also analyze the residual nitrogen sources present in the environment. So we analyze what is really used or not by the cyanobacterium. And for this part, so in blue, we can see that at the end, we still have 60% of nitrogen. So the cells was not able to use all the nitrogen that we introduced at the beginning of the growth. And if we analyze the nitrogen reserve, we can see that there is no significant difference in the content over the time, explaining why we are in a nitrogen excess condition. The second profile that we obtain is associated with the reduction of the growth. As we can see in this case, we reach only around 0.5 of optical density. We can see in blue that we have a complete assimilation of the nitrogen sources here. And we can see in pink and in purple that we have a degradation of the intracellular nitrogen reserve. So we are in nitrogen limiting condition. And these two profiles are associated with a different exopolysaccharide production. If we take as an example the nitrate conditions, we test 3 concentrations. And we can see that we have an increase here in exopolysaccharide production when we decrease the nitrogen content. Why? Because in this situation, you have a deficiency of nitrogen, but you have an excess of carbon, and this excess of carbon is associated with an important production of exopolysaccharides. The last condition that we tested, so the condition in which you have only the nitrogen of the atmosphere, was surprising because we can imagine that use the nitrogen of the atmosphere as a nitrogen sources it is using non-limited sources. So we can imagine to find something representing a nitrogen excess condition, and it was not the case. We observe a reduction of the growth and a complete degradation, as we can see clearly in pink, of the cyanophysin granules, and also a degradation of phycobilly proteins. So even if the sources seems to be unlimited, the strain is not able to use the sources efficiently, and we are in a condition mimicking nitrogen limiting condition. In this case, we observe an important production of exopolysaccharide, which is in fact the highest content that we obtain among all conditions tested. Then we go further and try to analyze the composition of the polymers in the different conditions. We analyze 2 different parameters. First, we analyze the presence of non-saccharidic constituents, or the sulfated groups and the proteins. But in a second way, we analyze if different monosaccharides or not are composing the polymer. Concerning the non-saccharidic constituents, you have here, so in brown and in pink, the proportion of saccharidic compounds, so sugar, and in purple and in blue, the non-saccharidic constituents. And you can see that proteins and sulfated group both are present in all conditions, but at different concentrations, in different quantities. And it is interesting to have these 2 non-saccharidic constituents because it will increase the complexity of the polymer, their physiochemical chemical properties, and so perhaps their use in industries. Then we analyze the monosaccharide composition, and what we globally observe is a modification of the monosaccharide composition according to the nitrogen sources. if we take the example of nitrate as a nitrogen sources, we have an important amount of exos, reaching more than 90% of the polymer. So we have a huge content in glucose, mannose, rhamnose, and galactose, represented here, so in blue, in brown, in gray, and in green. And we have a low content of pentose, arabinose, and ribose, represented here in orange. But if we use urea as a nitrogen sources, the profile is completely different. You have a decrease in exos, so a decrease of the blue, the green, the brown, and the gray monosaccharide, but an increase in arabinose and galacturonic acid. So we have a high proportion of, in this case, the purple, and the orange hexagons. So it is very interesting because just by changing the nitrogen sources, we can change the composition of the polymer, the physicochemical properties of the polymer, and so perhaps the sector in which it could be used for industries. And so the last step was to see if the polymers could be interesting or not for these industries. We test multiple possibilities, and 3 give interesting results which need to be further investigated. Use the polymer as bioemulsifying agent, for example in food industry. Use the polymer as antibiotics, and use the polymers as what we call a biostimulating agent. In fact, a biostimulating agent is a molecule which could help the good germination, the optimization of the germination process, the development of seedling, and which also play on the plant defense mechanism. So it's very interesting for agriculture. And I will focus on this example for the rest of the presentation. So effectively, what we wanted to do is, okay, if the exopolysaccharides could be interesting or not as plant defense inducers. For that, we first test in vitro, by in vitro assay, different exopolysaccharide solution on Arabidopsis thaliana cultures. And thanks to these preliminary results, we go further by testing under greenhouse experiments the solutions on tomato plants. And we analyzed 3 different parameters. First, what we call the phenylalanine ammonia lyase activity, the PAL activity, which is an enzyme. We then analyze the free proline content, so the content of 1 amino acid, and the content of 1 pigment, the anthocyanin. And why we selected these 3 parameters? Because they are all implicated in plant defense mechanisms. So it indicates that if you have an increase of these parameters, it could indicate an activation of plant defense mechanisms. Concerning the in vitro assays, we have here the cell culture of Arabidopsis thaliana. We introduce the different exopolysaccharide solutions, and then we analyze different biochemical parameters. We in fact analyze the enzyme activity, the PAL enzyme. We test different fractions separately. First, the RPS fraction. So here, the fraction which is completely free in the environment. And if we compare with the control, you can see that there is no difference in the enzyme activity. Then we test the CPS fraction, so the fraction still attached to the cell membrane. In this case, we have an increase of the activity, which is 8 times higher than the control. We test then the combination between RPS and CPS, so the total EPS. We also have an increase of the activity, which is 3 times higher. Then we test the CPS, but still attached to the cell, the Gloeotece verrucosa cells. And we also observe an increase of the PAL activity. So we test the cells alone. We have cells without any exopolysaccharides, and we observe no difference. It clearly indicates to us that the results obtained are associated with the CPS fraction of Gloeotece verrucosa. So the next step was to hydrolyze this CPS to see if the result was associated with the polymer itself or the monosaccharide pool forming the polymer. And when we applied the hydrolyzed solution on Arabidopsis thaliana, we observed no difference in the PAL enzyme activity. So this is the polymer itself and its structure, which is very important for the response of the cell. So, thanks to this result, we select the CPS fraction and the total EPS of GLOW-EOTC verrucosa as good candidates for the greenhouse experiments. And for that, we test to use these two fractions by two ways. First, as seed priming agent. So we soak the tomato seed in the different solution, and we analyze the germination process, the development of the seedlings over the time. What we observe, first on the left part of the slide, we have in orange and brown all CPS fraction tested but at different concentrations. And with the different shade of green, the EPS at different concentrations. We observe for the germination percentage that there is no difference according all conditions. So when we soak the tomato seed in the different solution, there is no difference on the germination efficiency. And when we analyze the length of the seedling at the end, we can see also that there is no difference concerning the size of the seedling at the end. So the polymer seems to have no impact on morphophysiological parameters. But when we go deeper in the investigation and we analyze biochemical parameters of the seedling, we observe a decrease of pal activity and an increase in free proline content on this seedling. So it indicates that even if there is no response at the morphophysiological level, there is a response of the seedling intracellularly, and it needs to be deeper investigated. Then we applied the solutions by a second technique, which was a foliar spraying technique. So we spray directly the solution on tomato leaves. And once again, we analyze different biochemical parameters. We observe globally that we have an increase in the three parameters, so the pal activity, the free proline content, and the anthocyanin content for the CPS fraction at a concentration of minimum 2 milligram per milliliter. And we have also a response of the plant with the EPS, the total EPS, but the response is less observed. So all these experiments lead to the conclusion that the CPS fraction of GLOW-EOTC verrucosa could be a good candidate to further investigate for application in agriculture. So to conclude this presentation and to sum up the thesis, we play with GLOW-EOTC verrucosa, we modulate the carbon-nitrogen ratio. We observe that we can optimize the exopolysaccharide production by modulate the CN ratio and create an excess of carbon by reducing the nitrogen content. We can also change the monosaccharide composition by changing the nitrogen sources. And we observe that the polymer could be interesting in different sectors of industries and certainly as biostimulating agent. Concerning the perspectives, there is two approaches. The first one is more associated with the fundamental research. Okay, we modulate the production of exopolysaccharides, but what we observe is that the strain is able to produce exopolysaccharide in all conditions. So there is a constitutive production of exopolysaccharides. And it could be associated as a role of protection. If we analyze the environment in which the cell was isolated, the strain was isolated, the rice field in India, there is high light intensities, UV radiation, and desiccation period. So this is harsh conditions for the development of GLOW-EOTC verrucosa, and the exopolysaccharides could be a way to protect the cells. Then concerning the perspective for the applications of exopolysaccharides, I think it's very interesting to go further concerning the biostimulating properties of the polymer, of the CPS fractions. And for that, we can apply different stress on tomato plants, a pathogen stress or a salt stress or a thermic stress, and see if when we apply the different solution, we have a response which is important or not of the tomato plants. But it's also interesting to test the polymer on all the crops or the vegetables or the plants. And finally, to test the polymer in a natural environment, so in the nature, to see if the response is the same as in the laboratory conditions, and also to analyze the effect on non-targeted organisms, fauna and flora. So I would like to thank all the members of the jury to be here today once again, but also thanks all the members of the lab and of Materia Nova for their help during these 5 years. Thank you very much.
Igor, c'est à toi. Il m'entend. Igor, tu m'entends? Igor, tu m'entends maintenant? Ah, voilà.
Igor, tu nous entends? Ton micro est coupé.
Il est loin. Hein.
Tu seras là allumé.
Ton micro, Igor.
Okay, so thank you very much, Camille. Do you hear me all?
Yes, yes.
Absolutely.
Okay, you hear me?
Yeah.
I don't hear you. Is that okay for me? For you? You hear me?
Yes, we hear you.
Okay.
Do you hear us?
So, Camille, thank you very much for this nice presentation. So, we probably, if or the expert, if know some other questions. I remember you that we had a meeting together, and we already posed a lot of questions to Camille, but maybe so the expert if some more questions, and I will give the floor to the person who is the farthest to Mons first, who is Philippe Michaud, Professor Philippe Michaud. So I give you the floor and see if you have some more questions and comments.
Yes, thank you, Igor. Do you hear me?
Yes.
Okay. In the first time, I would like to thank Rudy and Camille for their invitation to participate to this PhD defense. Dear Camille, thanks a lot and congratulations for this talk and for your revised manuscript.
Thank you.
I have only 3 or 4 questions. The first one is what was the motivations to work on the EPS from Glottis verrucosa during 4 years? It is only because it is a diazotrophic cyanobacteria with a mucous phenotype?
So the thesis was associated with the project named Algotech project, and the aim of the project was to have organisms in which we can find different high added value compounds, and my part of the work was to work on exopolysaccharides. So we select an organism which is known to produce exopolysaccharides, but for which there is no more information in literature. So working on something new. Just we know that there is exopolysaccharides, but no information. And we select effectively a diazotrophic strain because we imagine that it could be easier to produce a high amount of exopolysaccharides at a large scale with only CO2 and the nitrogen of the atmosphere as nutrients for the beginning.
Yes, okay. But this cyanobacteria strain was not chosen after a screening of cyanobacteria producing EPS?
No, it was only based on literature research.
Okay, thank you. The second question is the roles of EPS for cyanobacteria. You describe several roles such as adhesions, motility, protections, etc., etc. And at the end of your presentation, of your talk, you give the hypothesis that the EPS could help the cyanobacteria to to fight against desiccations. But have you other ideas of putative roles of EPS?
Yes, so in gliosis varicosa, it could be associated not only with hydration and dehydration cycle, but also associated with UV radiation. And so perhaps there is some pigments associated with UV phenomenon, which could be implicated in the exopolysaccharide matrix, such as the mycosporine amino-like proteins and cytonamine, but it could be also associated to a role in which the matrix of EPS could integrate different nutrients which could be deficient, for example, in the environment. And so in the EPS matrix, these elements could be more bioavailable for the strain.
Okay, but I'm not totally agree with your opinion, because the different cyanobacteria species have several EPS in terms of structure. You understand?
Yes.
So why the evolutions have has conserved in cyanobacteria very complex genes, very complex different genes depending on species, giving different exopolysaccharide with different structure and compositions.
Yes.
If the sole role is the preservation of moisture, it is not necessary to have the large diversity of structure currently observed in literature.
Yes, so that's why I think there is multiple roles which are possible, but effectively we can also imagine that according to the condition in the environment, the structure could be different. Even if it's a role of protection, the protection is not necessarily the same protection for the same problem, and so that's why the structure could be different.
Okay, and another question is why cyanobacteria such as glottis varicosa change the compositions of its EPS depending on the nitrogen nutriment available or other conditions. It is very complex to synthesize an EPS for cyanobacteria.
Yes, so I think that...
For example, sorry, Camille.
Oh, no problem.
Why with some nitrogen source, the cyanobacteria excrete an arabinose-rich polysaccharide and not with another nitrogen source?
I think that it's associated with the modification that the nitrogen sources could induce on the metabolism. Typically, urea is considered as a nitrogen and the carbon sources, so it will completely, the response of the cell would be completely different between urea and nitrate. So the carbon metabolism will be different, and the monosaccharide produced will also be different, explaining why the composition of the final exopolysaccharide will be also will not be the same in the different conditions. If it's associated with the carbon.
Sure of that, did you examine the metabolic pathways and the anabolic pathways leading to the biosynthesis of all this monosaccharide?
It was one of the aim, but we were not able to have a good lysis of the cells. But effectively, it was one of the aim to analyze in the different condition, see what could be different in the carbon metabolism and in all the pathway associated with monosaccharide production, glycosyl transferase, and also polymer export. So I think that even in literature, there is no clear response concerning this production of exopolysaccharide and how it clearly works in cyanobacteria. But in my opinion, there is clearly an impact of the pool of monosaccharide present in the cell will clearly impact the exopolysaccharide which is synthesized at the end. If it's associated, for example, with an excess of carbon, as in our condition, we can imagine that according to the condition, the nitrogen sources, the excess of carbon will not be associated with the same monosaccharide, and so perhaps the monosaccharide introduced in the polymer will be different.
Yes, but be careful before to conclude with the variability of these compositions. I agree with you because in literature, numerous authors describe the variations of EPS composition depending on the culture conditions. But as you know now, the biochemical investigations for EPS is very difficult, and the purity of samples and also the hydrolysis conditions could
influence
give artifact and could give to not good conclusions. And as you see in your revised, as I seen in your revised manuscript in the figure 4.9, your sample of EPS and CPS have low sugar content.
Yes.
We have discussed about that during the first defense. And I think the better purifications of samples could be necessary to conclude, really.
Yes, I'm absolutely agree with you. That's the conclusion of this part.
But you are very honest in the revised manuscript. It is
yes,
a good thing.
That's
next question for you is once again is the frontier between release polysaccharide and capsular polysaccharide. For me, the frontier between these two families of EPS depend mainly on the extraction procedure. And do you think that if you use another extraction procedure, you will have the same RPS and CPS ratio?
Yes, I think that there is effectively an impact of the extraction process. For example, just filtration or centrifugation of the culture will impact, I think, the content in RPS. And concerning the rest, so the CPS, I think that also there is an impact of the protocol of extraction of the CPS. So yes, there is an impact. I don't know if there is an effect on the composition for the RPS part, but there is clearly an impact on the content that we can have.
Okay. To conclude, do you hear me?
Yes.
Yeah. To conclude, I think that your perspective of industrial applications of EPS from cyanobacteria are very optimistic.
Yes.
Could you give to us the interest for industry to work with EPS from cyanobacteria versus those of terrestrial plant, seaweed, or some bacteria?
Yes, concerning plant, I think that it can reduce the use of arable land. For example, if we have
Camille, sorry, excuse me, but what is the part of landing plants of crops used to produce polysaccharide?
To be honest, I have no idea. But I suppose that
It's a very minor part for polysaccharides such as guar or caru, for example, and for others such as pectin. They are extracted from byproducts, so it is not a problem.
Okay, yes. I think that cyanobacteria could be organism in which the nutrients entrance could be reduced in terms of quantity if we use, for example, the CO2 or the denitrogen of the atmosphere. So it could be a positive point at this part of the process. And if we compare, for example, with other bacteria, there is also a difference in the complexity of the polymer, and so perhaps in the application that we could have at the end with the polymer.
Okay. For me, I will conclude with that. You have you have two challenges to permit to EPS from cyanobacteria to go on the market. The first one is to have a progress in structure determinations, in full structure determinations, to go to correlate this structure with biological functions, for example. It is very hard to do that. And the second challenge is not really a biological challenge, but a process challenge to lead to high productivities in EPS.
Oui, yes. Yes,
Okay.
I agree with this point.
Finish for me, Igor. Thank you.
Okay, thank you, Philippe. So the second expert external to the University of Mons was Professor Konrad Meulert. So I give the floor now to Konrad. If you have some questions, some remarks or comments.
Okay, thank you, Camille. I would like to start with to congratulate you.
Thank you.
And also with a very nice presentation. It summarizes your work very nicely. You probably experienced quite some challenges.
Yes.
You mentioned it was not easy to grow the algae, and you probably had to learn several new analytical techniques, experimental techniques, to test the activity of the polysaccharide. But that is the aim of the PhD.
Yes, absolutely.
And then another aim of the PhD is that some experts can ask you some questions, so I have some questions for you. So it was difficult to grow the species. Did you try to grow it as a biofilm, or would it maybe grow better as a biofilm?
We try effectively to reduce the agitation speed to see the formation of biofilm. So there is a good growth of the strain in biofilm. And I think it's a characteristic of the strain, but the extraction process was very complicated in this case. It's already difficult when it's with an optimal speed, but without any speed, it's complicated. So to recover the exopolysaccharide, it was very complicated, and there is also a lot of biofouling on the flask. But definitely, I think that the biofilm is an important process in the growth of the strain.
Okay. You manipulated the nitrogen supply, and usually it has a very big effect on the cyanobacteria. You mentioned that the species forms phycobilizone proteins. It has phycoerythrin and phycocyanin. Did you see a change in color of the culture when you reduce nitrogen supply?
Yes, we observe a change of the color, so it switched from green to yellow, so it was more bleaching, and I think associated with the end starvation condition. So we did not see a clear change in the color, but just the green was light and it was more yellow. Yes.
Did you also see increases in glycogen?
We did not measure the glycogen content, so I have no idea, but I think that in case of light direct cycles, there is a variation of the glycogen content over the light direct cycle, but in case of continuous light condition, I have no idea. I suppose that perhaps the cycle is still present at the beginning, but on a long-term experiment, it was asked for the problem of the diazotrophy. We have no reference and no idea of what's happened on a long period.
But it's impressive the changes you see in the polysaccharide composition.
Yes.
In terms of sulfate groups, in terms of protein content, but also in terms of monosaccharide composition. Very different. Did you check the reproducibility? So if you would do the experiment several times, do you get the same, under the same conditions, also the same monosaccharide composition?
So we test 3 biological replicates, but
Yeah.
at the same time,
Same time.
yes. But I did not reproduce the experiment twice or third times to be sure.
Okay, but you probably grew the culture many times to produce the polysaccharides.
Yes.
For the experiments with the plants, for instance.
Yes.
But you never reanalyzed.
Yes, I performed because in fact the 2 experiment was performed at the same time. So I optimized the HPLC method before the plants. I observed a profile which was the same after the plants experiment because the rest of the experiment was performed. So normally it was the same during the plant experiment.
Yeah. Okay. Did you see any unknown sugars in the chromatogram, or
No.
every peak matched very well with the standard?
With the standard, yes.
Because some, for instance, in spirulina or Prospira or Limnospira, there's a quite well-studied polysaccharide which is called spirulina. And
Yes.
that polysaccharide, they identified some very unusual sugars. So
Yes.
you might expect some unknown sugars or very rare sugars in extracts from other cell bacteria.
Yes, but I suppose that also if these special monosaccharides are present in low content, perhaps because we need to optimize the process, we could have the observation of this monosaccharide when the process will be optimized. Because we observed that the total compound the total quantity that you have at the end was not very high. So perhaps we missed some information.
Did you think of looking at the rheology of the polysaccharide? Or?
We wanted to do, but we didn't have the time to perform this, but I think it's very important,
Yeah.
yes. And the viscosity is also very important, yes.
Do you think it could be possible? I think that the species produces a lot of polysaccharide. So in some
Yes.
conditions, 0.4 gram per gram of biomass, that's a very high productivity. Do you think you could stimulate even more by genetic engineering of the species? Is that something?
Why not? I think it's a possibility if we manage to have a very good lysis of the cell and we better understand the metabolic pathway implicated in the exopolysaccharide production. Why not play,
Yeah.
yes, on mutants and
It
so.
would be a possibility to fine-tune the monosaccharide composition? Is that,
It's complicated because I think we don't know if the polysaccharide is produced with the excess of monosaccharide present in the cell, and so the excess presence at the T time is associated with the polymer, if there is a specific production of monosaccharide to produce the specific exopolysaccharide that we obtain at the end. So if we have the answer to this question, it could be a possibility. If there is a synthesis of specific monosaccharide for the polymer, we can play on it and specifically induce monosaccharide. But if it's associated with the excess present in the cell, I think it will be more difficult to have a prediction of what will happen.
do you know if there's already a lot of research in this area in other species or maybe even in other prokaryotes?
For the analysis of
The composition?
I think that in Synechococcus and Synechocystis, they mainly play on that, but it's not associated to change the polymer structure. It's more associated to see the role of the exopolysaccharides.
Yeah.
So it's not really associated to produce something interesting for industry.
So if you would produce it for the industry, what a biostimulant, a plant biostimulant,
Yes.
would be an interesting route. What is the regulation? Is it complicated to get it on the market once you can produce it?
Yes.
Is there a lot of regulatory issues?
I'm not sure. Yes, there is, but it's not really clear, because when we check on different websites of company which produce biostimulant, there is no clear information. So I think that yes, there is regulation, and mainly in Europe, but it's complicated to know what kind of regulation.
And would you then go for a pure extract, which is maybe more difficult to produce, or can you use the biomass as it is?
I think it could be cheaper to use the total biomass, but we need to be sure that there is no interference with other compound of the biomass, and see if the CPS fraction in the biomass, which seems to be interesting for us, it's in enough content with all the rest of the biomass to have an interesting result at the end. So we could test it that, or only have, for example, cell associated with the CPS. Why not test this possibility? Perhaps not all the extract, but cell with CPS could be interesting also to test.
Okay, thank you. These are my questions. And congratulations.
Thank you.
Okay, thank you. Thank you, Conrad. And our next expert was internal, this time to the University of Mons. It was Professor Annelise Sanson. So Annelise, do you have some questions?
Yes, I take the microphone. Do you hear me? Yes? No?
Yes, yes.
Yes? Okay, it's nice. Try to. Okay, thank you, Camille, for this very clear presentation. I think that you give us a very global view of your works you have done during these 5 years for the Algotech project, with some difficulties for the culture of your cyanobacteria. And in this way, I have some questions for the culture, because you always realize your culture in liquid medium. So
Yes.
do you think that a complete change of technology should give more grow, but not grow rate of your cell, but easier way to collect the exopolysaccharide? So I think that. Do you think that using immobilized reactor
Yes.
should give you some more result, easier way to collect exopolysaccharide?
I have no idea. It could be interesting to test. I think that everything could be interesting to test in the aim to have a better production of exopolysaccharides, but it's difficult to know because there is not a lot of information about that. So
because with this kind of reactor, you have the opportunity to use nitrogen coming from atmosphere.
Yes.
Easier because you have best exchange.
Yes.
Atmosphere. You show us that you have limitation when you use nitrogen coming from atmosphere with liquid traditional culture.
Yes.
And I think that the problem is the dissolution rate and the solubility of nitrogen in your medium. So it's not so easy to have the concentration you need for the growth of your cell when you use directly gaseous form of nitrogen. So I think that using immobilization with say that in English, biofilter technology may improve your productivity as well in terms of cells, but also in terms of exopolysaccharides.
Yes.
So have you tried to produce and to cultivate your microorganism on a petri dish?
Yes.
Other nitrogen?
Yes, only with the nitrogen of the atmosphere, yes. And the growth was not so good as when we introduce the nitrogen sources in the plates. So we have a growth, but the number of colonies in the plates was very low, and it takes a long time to have a good plates.
So you show us also that your polysaccharide has a lot of sulfated groups, and we see that the percentage of sulfated group is sometimes very high.
Yes.
Have you tried to do the balance, the mass balance between the sulfate you have in your medium and the sulfate you obtain in terms of quantity? Because it was very important.
Yes. No, I didn't check, but effectively the content is very high. It could be interesting to compare with what we have at the beginning. Yes.
Do you follow in your medium the consumption of sulfate?
No, we only manage the nitrogen and the phosphate for the NXS condition. But yes, it could be interesting to see because there is no, it's a poor medium at the beginning. So we can imagine that effectively there is something. Yes.
Restriction in the, if you need some production of exopolysaccharides for the grow because it is necessary for the metabolism or for the protection of the cell, perhaps you have a deficit of sulfates in the medium. So perhaps you can help and can increase the grow and the concentration. Just for the objective to use this kind of exopolysaccharide in industrial area, what is the dry weight you have with an optical density of 1?
The dry weight, the correlation is around 1, so it's like for Arthrospira. It's relatively the same. So.
Because I think it's not a high quantity.
No. No, the optical density is not high, so the biomass it's not really high also.
Last question concerning the application for biostimulation of the plants. You said that you use directly the exopolysaccharide as well as the capsular or the release, but do you try to hydrolyze but not completely your polysaccharide to obtain some oligomeric polysaccharide and know if it is better with smaller than this very long polysaccharide?
No, we didn't do this, but effectively it is interesting because in many studies they highlight the positive effect of oligosaccharides on plant defense mechanism. So yes, I think it's an interesting idea to test. But for that, we need effectively to have an idea of the efficiency of the hydrolyze according to the condition that we test to have something different sequence and see if we can have different pool of oligosaccharide. Yes, it could be very interesting to do that. But it's also interesting because when they discuss this in literature, they always discuss about the phyllosphere and so the organism present on the surface of the leaf. And one hypothesis is that even you have important polymer, there is a degradation of this polymer by the phyllosphere, and so this is oligosaccharides which can react with the receptor of the plant and could induce the effect on the plant. So it could be interesting to test.
Last question: do you think it will be possible to separate the growth and the production of exopolysaccharide, because it will be easier for the management of the culture if it is possible to separate these two steps of the production? Do you think it's possible with your cell?
Perhaps yes, with work by a first stage of increase the optical density with a high nitrogen content, and then switch on a medium in which we reduce the nitrogen content to have a high production of exopolysaccharide. I think it could be possible to test that. Yes.
To facilitate.
The recovery of the cell and in the supernatant. Yes,
if you have
more cell, we have more exopolysaccharide. Absolutely, yes.
Thank you very much. Congratulations.
Thank you.
Okay, thank you, Annelies. So we went around all the experts now. Professor Rudy, what you was the director of the thesis. Rudy, do you have a comment to do now or
no,
later?
I have no comment.
Okay, so it's a tradition to pass the word to the audience. So if somebody in the audience wants to pose a question, so it's the time now. Et on peut le faire en français aussi, évidemment. Donc si quelqu'un veut faire un commentaire ou une question, c'est le bienvenu et c'est le moment de le faire.
I have a very applied question. So you have some very interesting results in the natural defense area. So, how did you store your samples? Did you freeze dry them?
The sample of exopolysaccharide or the plant? The exopolysaccharide, we extract the exopolysaccharide, then we lyophilize the exopolysaccharide and we keep it at room temperature. And then just few hours before the experiment, we add the water to prepare the solution. Not so easy. You can ask to Charlotte. It was not so easy. It's difficult to have a good hydration when they are lyophilized.
Do you think there is something you can do to make it more soluble? Because I'm asking this because if you want to use this in a professional setting, you need to make a formulation of this.
Yes.
It's better if you have a dry product that you can formulate.
Yes, and then yes, that's a good question. I don't know. I have no idea, but yes, it's a point in which we have to work on. Absolutely.
Well, okay. So normally we have to group together, so we will do that virtually. I don't know how. Je sais pas trop comment. On se voit comment alors dans une salle virtuelle ou?
On va rester nous ici dans le Teams. Et donc là vous devez vous connecter via le Teams. On va faire sortir l'ensemble des gens. Et nous on va se réunir en Teams maintenant.
Ouais, c'est ça, voilà.
Ok, super.
Ici, comment on coupe le fond d'écran? Et on met no malheureusement.
On n'a pas récupéré les doigts encore.
C'est loin, Madagascar.
Voilà. Normalement, tu devrais voir maintenant. Tu devrais nous voir, Philippe.
Oui, je vois Camille derrière,
Voilà.
avec son masque.
On va approcher là.
Est-ce que vous m'entendez? Oui, je pense. Je vais me mettre à côté de Camille. Donc voilà, je vais officier puisque Igor ne semble pas
C'est très bien.
nous rejoindre. Donc voilà, je vais vous proclamer les résultats. C'était pas... L'attente était peut-être longue, mais je pense qu'il n'y avait pas de suspense. Donc, je suis censée lire le texte. Vu le décret du 7 novembre 2013 définissant le paysage de l'enseignement supérieur et l'organisation académique des études, nous, présidents, secrétaires et membres du jury chargés de conférer le grade académique concerné, déclarons que Camille Van Camp, née à La Louvière le 13 octobre 1993, a obtenu en l'année académique 2021-2022 le grade académique de docteur en sciences. Félicitations.
Merci.
Tu fais ce que tu veux.
Voilà. Je vais céder la parole au promoteur. Je pense qu'il a quelques mots à dire.
Oh, je ne sais pas, j'ai rien préparé du tout. Hein, donc, il y a deux choses qui, dans cette thèse, qui caractérisent en fait très bien cette thèse et son parcours. D'une part, c'est la première fois que j'ai un couple qui a, je veux dire, commencé une thèse et qui a réussi à la finir sans se disputer. Et qui sont toujours ensemble apparemment. Et donc ça, c'était une première pour moi. Au départ, quand j'ai accepté, je me suis posé quelques questions et c'était pas si évident de cela d'accepter, parce qu'on connaît un peu la nature humaine. Et en tout cas, je sais pas comment vous faites, mais en tout cas, moi, je vous admire. Rien que déjà pour ce point-là. La deuxième chose qui caractérise son parcours, c'est son opiniâtreté. C'était pas un sujet facile, c'était un sujet qui n'était pas simple pour diverses raisons techniques, et particulièrement que cette cellule ne se laissait pas faire et que plus d'un en tout cas se serait découragé et aurait envoyé, je veux dire, les cultures par-dessus les fenêtres et m'aurait dit, allez, le sujet est vraiment un sujet de... Mais bon, c'est vraiment quelque chose qui te caractérise. Et la troisième chose, et on en parlait tout à l'heure, je veux dire, lors de la délibération, qui te caractérise aussi, et c'est probablement une des choses les plus importantes pour moi, en tout cas pour un chercheur, c'est l'honnêteté intellectuelle. Tu as une honnêteté intellectuelle qui, je veux dire, est vraiment une des valeurs sur lesquelles on doit s'accrocher, sur lesquelles tu dois t'accrocher. Qui, à mon avis, fait de ta personnalité, mais qui est vraiment toi. Et cet intellectuel, je veux dire, cette valeur-là, il faut la conserver. J'espère que tu la conserveras, je veux dire, longtemps. Maintenant, on a beaucoup parlé de métabolisme, on a beaucoup parlé de division cellulaire, mais ton métabolisme est en train de changer.
C'est clair.
Et en tout cas, tu vas maintenant commencer une nouvelle aventure qui est probablement la plus belle des aventures, je veux dire, pour un humain. Et moi, je te souhaite et je vous souhaite, je veux dire, vraiment de vivre cette aventure pleinement et de prendre le temps. Je veux dire, je crois qu'il y a une caractéristique qui est en train de nous échapper dans le monde, c'est prendre le temps de. Et je crois qu'il va falloir que tu prennes le temps de.
Oui, ça me fera du bien.
Et en tout cas, toutes mes félicitations.
Merci beaucoup. paraît que je dois faire un discours, du coup. Allez, quelques mots pour faire plaisir aux gens qui se sont déplacés pour ça. Je voulais d'abord remercier vous, chef, pour m'avoir permis justement de faire cette thèse. Même après l'échec du FRIA, vous avez quand même eu la brillante idée de me garder. Ou vous êtes un peu fou, je ne sais pas, peut-être un peu des deux. Mais en tout cas, je voulais vous remercier de m'avoir donné cette opportunité. Je voudrais remercier aussi tous les membres du jury d'avoir pris le temps de lire mon travail et de l'avoir amélioré, chacun dans son expertise. Donc pour ça, je vous remercie beaucoup. Je voudrais vous remercier aussi particulièrement Anne-Lise parce que vous m'avez aussi suivi pendant tout le projet Algotech. Vous m'avez vu désespérée à toutes les réunions et vous avez toujours essayé de trouver une solution. Donc merci beaucoup pour ça aussi. Je voudrais aussi remercier tous mes collègues qui sont là ou en ligne. L'aventure d'une thèse ne serait pas la même si on n'avait pas des collègues sur qui s'appuyer et avec qui passer du bon temps au labo. Et clairement, sans vous tous, ça n'aurait pas été pareil. Vous allez vraiment me manquer. Et je vous souhaite le meilleur à tous. Augustin, Aïcha, Alice, I will see you soon for your defense. Je voudrais aussi remercier les amis et la famille qui se sont déplacés aujourd'hui. Je sais que ça fait 5 ans que vous entendez parler un langage que vous ne comprenez pas nécessairement. Je vous promets, c'est terminé. Mais merci de m'avoir écouté et d'avoir essayé de comprendre mes problèmes et mes joies, même quand ce n'était pas toujours très clair. Et je voudrais terminer par remercier Guillaume, sans qui, clairement, je ne serais pas là aujourd'hui. Tu as été aussi le moteur de cette thèse. Clairement, sans toi, je n'aurais pas terminé aujourd'hui. Et je suis ravie de me dire que tu auras la même force et le même courage pour notre fille. Donc voilà. Je pense qu'il est l'heure de vous offrir un verre maintenant. Merci à tous.
Un tout grand merci Philippe d'avoir été parmi nous et j'espère qu'on aura l'occasion bientôt de se voir. Un tout grand merci.
Merci beaucoup.
Merci à tous. Au revoir, bonne soirée.
Merci beaucoup.
Félicitations.
Merci.